semester, I have taken the bonus quiz from the students. So that bonus quiz actually taken from the last lecture. The last lecture is the population genetics, and most of the question for the bonus quiz, I combined population genetics and also the sex the sex determined chromosomes. Okay, or the X-linked chromosomes. What is also the ploidy? Yeah, polyploidy. What is polyploidy? So this kind of question, I remember I make from this 2 to 3 lecture, I think, and then I have taken some kind of the bonus quiz. But this semester was very short. I couldn't provide all the lectures that I supposed to give you. Alhamdulillah, so far I have done 14 week with 10, I think 10 or 9 or 10 lecture, because this is the genetics. Some of the lecture is given within 2 weeks, if I'm not mistaken, at least 2 lecture or 3 lecture like that, right? So I will not be conducting the bonus quiz because, as I promised, I'm not going to conduct another lecture for the next week, which is the population genetics. If you want to, take the bonus quiz, but I think that it is not really necessary because the grades are perfect. I mean, the grades, everyone wants A now, and it will be hard, really. So the question for the exam, this is the tough season is coming for the exam, because I have provided like how much, like 40 is remaining or 50 is remaining, 45, right? 45 is remaining. So we will do in the final quiz. So you have a long week rest, like 2 weeks. We have final exam. It is on 19th of May 19, right? May 19, we have final exam. So you still have a long way to prepare. And I will not conduct the bonus quiz because reason being, I'm not giving the last lecture. If you want to do here the last lecture, maybe next week we have to conduct the lecture as well as the laboratory part. Okay. If you don't want, you want to stop by here and you don't want to take the bonus quiz, then we will just stop by today's lecture. And for the sex-linked inherited disorders, we have to do a laboratory session this week, inshallah. Is it clear? Stop again. All right. So 1 minute left for 15. Okay, let's start. Okay, so we are reversing back to the molecular genetics, okay, not the classical genetics. We are reversing back to the molecular genetics, and from the molecular genetics, what you have known that there is a central dogma. That central dogma is consisting of 3 types of principles, right? What do you call it? The downgrade from the DNA to RNA, from RNA to the protein or amino acids. So this kind of chain is always downgrading this way. Yeah, we can reverse it back. Actually, we can reverse it back, but for genome, for the DNA sequence, from the DNA sequence, we can have some rough idea, but we will not have any knowledge about the final product. For the final product, we have to study about the RNAs and how RNA convert to be a polypeptide chain. Yeah, polypeptide chain. So there are some RNAs, different types of RNAs involved in translation, right? But today's lecture is a little bit different from those kind of central dogma I will consist some of the principles to here, but before going to the central dogma, we need to distinguish what is the eukaryotes and what is the prokaryotes. Are they same? I mean, in terms of the gene that they code, yeah, are they same? Because all we have nucleus on the, or all we have the genetic sequence, the DNA sequence, right? But what is the difference between organism to organism? I mean, why we say that there is a eukaryotes and there is a prokaryotes, right? So eukaryotes is consisting all the complex, the different organisms, like including the human. But for the prokaryotes, we have to group out from the eukaryotes. So today lecture, I will start. I will talk about mostly about bacteria and also the bacteriophage. If you heard about it, bacteriophage is a virus. Yeah, that virus that consumes the bacteria, or the virus that is using bacteria cell to become to masking. Yeah, that's why virus. When we hear about virus, we are very like, oh, we are on alert, right? Like what happened to the coronavirus. So if you are interested how the gene is structured in this kind of prokaryotic cells, we need to understand some principles, some principles that is really regarding the bacteria and viruses. Okay, so when we hear about bacteria, what we think? Our resemblance, or what we think is, it is kind of a dirty, right? It is kind of a dirty place, or it's kind of a contaminated, dirty surface of something. But I do want to mention that many times all bacteria, they are, they are lumped together as a bad thing, right? As a stereotypical thing. It's about a bad thing, which shouldn't be happen actually. From the geneticist point of view, we should not categorize the bacteria as a bad things, because not all bacteria are so bad. In fact, many types of bacteria, they are helpful for organisms and ecosystems. So if you know any kind of this bacteria, maybe you can call it out to me, or you can just eager to know what kind of bacteria am I going to speak in today's lecture. So some of the bacteria, they usually that colonize your skin, that is beneficial, right? Some of the bacteria that colonizing our skin, that is actually useful bacteria or beneficial bacteria. That bacteria in our digestive system, they actually help break down food, and some can produce even certain vitamins. Okay, so the some types of bacteria, they are used in producing even some foods that we can eat, like lactose. Yeah, like a lactose, it is actually made of using the bacteria. So in ecosystems, bacteria have very important role as decomposers, and that is true not only the insect or microorganisms, but also bacteria. So the bacteria also, they have major roles in the nitrogen cycle. In order to do nitrogen fixation, that plants they need. See, here is again not only the microorganisms or in some particular insects. Here also bacteria can play important role for the as a decomposers. Okay. So these are just few examples of helpful and beneficial, yes, Ahmed, bacteria. So some not so beneficial, of course. That kind of bacteria are the cause of tooth decay, for example. Some forms of pneumonia, if you heard about it, that liver get infection, that is pneumonia, is caused by not virus but the bacteria. Also diarrhea, salmonella, and cholera. Yeah, cholera, when you have the neck problem in your neck inside. So that is due to the cause of the bacterial things. So we treat those things with what? Antibiotics, right? We take antibiotics and in order to kill those bad bacteria. So the antibiotics, they can be used to combat some of these related disorders, or the fatigue, or what you call it, the disease, yeah. On the other hand, some antibiotics are very important for destroying bacterial infections. We shouldn't leave out saying that some broad spectrum antibiotics can also harm good bacteria. So we shouldn't take for granted and take overdose of the antibiotics kind of things. That is why the doctor is prescribing some kind of antibiotics. We should ask, yeah, how often do I need to take, or doctor will prescribe it. If too much, then you can say, what about my good bacteria? It also can be that, right? So now, I also should mention that antibiotics do not work on viruses. Then, in that case, we have to take antiviral kind of medicines. Okay, why? Because the viruses, they are the pathogens that they are not made of cells at all. So here is the juice is coming here, that the what is that? The viruses, they are having no cells. Why? So I will tell you why. Additionally, there are some vaccines which can prevent many types of both bacterial and viral infections. So some of the vaccination, why we need to take some vaccination, because it can be universal type. It can kill against the bacteria, because the virus, what I say is, they have no cells, so they use bacteria in order to mask. Yeah, and the viruses can use bacteria, I will tell you. Okay, so what are bacteria exactly? They come in different types and different shapes, and also different size. Okay, some of them are, we call it heterotrophs. Heterotrophs meaning they consume or feed on some organic matter. Okay, and some bacteria, we call it autotrophs. Autotrophs, they can make their own food. Yeah, so it's like a chef. They can make their own food. Maybe these are good bacteria. So bacterium is just singular word. But when we are saying bacteria, that is a plural word. Okay, I will tell again. Bacterium. Bacterium, it is a singular form. But when we say bacteria, it is a plural form. Okay, so in today's lecture, I will overview of the genetics of the bacteria and what is bacterium. Bacterium is a prokaryotic cell. That means bacteria do not have a nucleus. If you remember from the beginning, yeah, I say they don't have a nucleus, or other membrane-bound organelles like endoplasmic reticulum, ROF, and smooth. They don't have this. But we will find ribosomes. We will find cytoplasm. We will find cell membrane. And nearly all bacteria have also cell wall. Okay. So like all living organisms, bacteria have also DNA. Yeah. So bacteria DNA, while still they are only double-stranded. So it is arranged in the circular form. From the beginning of the lecture, I talk about it. So how they are look at it. Yeah, they look very strange actually, but they are usually circular form. Depending on the species, some bacteria can also have flagellum. Flagellum is like a tail. If you look at the closely to the bacterial image, in order to help for them to move, and also they have a capsule which can help give them extra protection or give them attachment abilities. They will also sticky, I mean they attach, which can help with attaching to any kind of surface, including also each other. They also can, attach to each other. So in many bacterial species, they have a plasmid. Okay, because they don't have a nucleus. So, which is basically like extra DNA. So they have a circular, one very simple chromosome DNA, and along with that, different what do you call it, plasmid. Yeah, that plasmid have also their own DNA. Okay. So unlike our own body cells, which perform mitosis and cytokinesis, they also divide. So bacteria generally, they multiply even faster. They multiply very fast in a process we call it binary fission. Binary fission, okay? This is type of a asexual reproduction. Uh, when the bacteria can easily divide to make a copy of themselves, like a cloning, yeah, copy of themselves. So it is a asexual reproduction. Since it is an asexual reproduction, they produce what? Daughter cells that is identical would be expected to be identical to the parental cell, unless there is a kind of mutation. If some mutation happen in the bacteria, we say that they are divided not through the mitosis, it could be the meiosis, okay? So some types of bacteria, they do have the ability to share genetic material with each other because they are clone, yeah? So, however, because bacteria are a little simple in terms of genome size means in chromosome size, overall chromosome size, yeah? And structure, they are very good starting point for understanding some of the complexities of life. So bacteria can cause major disease of plants and animals as well. But there are also many species of bacteria that they are beneficial. And I told you some examples or some of the beneficial like a gut bacteria, skin bacteria, and there are so many bacteria that we consider as a beneficial. Okay. So what are bacteria? They belong to prokaryotes. Unlike plants that they are usually what? Eukaryotes, right? So bacteria usually single-celled and usually just have one single main chromosome. How many chromosome? One single main chromosome. That chromosome is not like in a eukaryotic cell X and Y. Yeah, they is actually circular form. That is meaning DNA is in a circular form. Whilst meiosis may not exist in bacteria, usually through the mitosis cell division, and recombination sometimes can occur, and genetic material also can be exchanged between different bacterial species and strains. So there are some particular strains and species of bacteria, they will exchange the genetic material with each other. So bacteria is the simplest living organism, and they have survived on Earth for the past 3 billion years. 3 billion years. So the bacteria have been found growing in all parts of the world, and they also play important role in ecosystem as a decomposers, yeah, or nitrogen fixation bacteria. So they tend to single-celled organisms, but some of them can be a bit more complex. So our understanding of prokaryotic genetics and molecular biology has been greatly helped by the understanding of the genetics of bacteria. I will tell you why. If you hear about the genetically modified organisms, actually we use what? Agrobacterium tumefaciens, right? Agrobacterium tumefaciens. So for the GMO crops and GMO organisms, we usually exploit bacteria. So bacteria can be considered to be haploid. Why? Because they contain single chromosome, but they have evolved other mechanisms for gene transfer and recombination, which I will explain in this lecture today, what kind of gene transfer and recombination do exist in bacteria, okay? So apart from the main chromosome, bacteria also have smaller circular DNA molecules. We call it plasmid. Yeah, we call it plasmid. So they also replicate and they also can be passed to the next generation. So these replacements can be quite small or can be almost as big as bacterial chromosome. I'm talking about the plasmids. What is plasmids? Yeah, why do we need to pay attention to plasmids? Now, at the end of the lecture, hope you will have a brief, some idea. Okay, let's move on. Let me check the chat box. The slides are not moved. Can you move the slides? Now it is moving? Yeah, it is moving, right? Okay. So the bacteria divided by what I say, binary fission. Let me type here. Bacteria divide by binary fission. Okay, by binary fission. Never forget this. So what this means, this is during the cell cycle. During the cell cycle, the chromosome is replicated, okay? And then one copy of the chromosome is partitioned to each of the daughter cells. So I'm talking about mitosis. So, but we cannot call, in case of bacteria, we cannot call this as mitosis. We have to say it is a binary fission or binary fission, okay? So if we inoculate bacteria, for example, on the growth medium, where we inoculate? In the growth medium, yeah? In a petri dish. With a single bacterial cell, they will start to grow and divide. So this is through the mitosis, but usually we call it binary fission. So more slowly at first, what happens? During this, we call it lag phase, yeah? lag phase shown on the slide here. And then they reach on exponential phase, where they rapidly divide and start multiplying. So in ideal growth conditions, for example, if we are feeding the bacteria or giving the ideal optimum temperature, so bacteria such as E. coli, yeah, everybody knows what is E. coli, it is harmful. They can double in number every 20 minutes. Every 20 minutes, they double. So within a closed system, things like a flask or media or in the even petri dish, for example, the bacteria will eventually start to run out of the nutrients. And what happens? The growth rate starts to slow down until the number of cells reach the plateau. When all of the nutrients are exhausted. So that this plateau stage, it is actually termination stage for the bacteria, okay? Or they have grown sufficiently, they achieve their mission, and that's it, okay. Okay, so bacteria, we can't really see by the naked eye, although we can see them under microscope, right? Or electron microscopes, where it is possible to calculate the number of bacteria. So we can actually, from the petri dish, we need to calculate what is that density, right? Density, even how many number. We can calculate the number of bacteria or the density of the bacterial solution by something called serial dilutions. So if they are very dense, we can just make a dilution, right? So this can also tell us whether their cell within a solution actually viable or not. So bacterial cell grows. The liquid medium can be quantified by transferring the neck and aliquot of them onto solidly semi-solid medium on a petri dish that is shown in the slide here. So the following, what we do, incubation and many divisions, usually overnight, it happens rapidly, right? So each cell gives rise to a, we call it a carni, that is visible on the surface of the medium. So this picture here shows the result of plating. The plating is serial dilution series, yeah? X to 10 times, or maybe the solution X to 200 times, something like this. So each dilution varies by a factor of 10. Okay, usually by a factor of 10. So in this case, each of these colonies, we call it colonies, that is visible on the spot, they are derived from a single bacterial cell, and we should say that there it is. So, but normally when you try to count them, what we have to do? We have to do this dilution series and then find out which plate has number that we can more easily count and we can calculate how many bacteria we are present in the undiluted sample that we started with. So we will compare from the initial bacterial statement by after dilution how, yeah. So this is usually done by pathologists or bacteriologists, virologists even, yeah. So they do this kind of work very, very often. So as I mentioned, bacteria contain single main chromosome plus one to several extra chromosomal DNA molecules. We call it plasmids. So these plasmids, they can replicate independently of the main chromosome compared to the main chromosome. And then they will get separated into daughter cells in the same way as bacterial chromosome can. So exactly same as a chromosome. So the most plasmids, they are dispensable to the host. So this means that they are not essential to the survival of cell. Okay, so in certain cases, these plasmids, they are necessary to grow in certain media, for example. So for example, if antibiotic was present in the media or on the environment, bacteria might have an antibiotic resistance, right? We call it antibiotic resistance chain on a plasmid. So without that gene on the plasmid factory, that would not be able to grow even whatever we do, what kind of effort we do. So similarly, bacteria need to often we call it the, what is that, ilicotent catabolize different types of chemicals. And sometimes these chains are present on these plasmids. See the role of the plasmids and how they are different from their normal chromosomal DNA. So bacterial chromosomes, they are much smaller in terms of size of chemical, much smaller than the eukaryotic chromosomes. Much, much smaller. Maybe 100 times, maybe 1,000, even the billion times of smaller than eukaryotic chromosomes. And they can range from about 130,000 base pair only, 130,000 base pair only, up to 14 million base pairs. That is why we say that bacteria, they are different size, different shape, even different genome. Yeah. So now let me talk about the plasmids. So plasmids, they are usually much smaller, ranging from his smallest 1,000 base pair only. That can be up to 200,000 only in terms of the base pair. Yeah, AB, not AB, AGCT, TT, GC, something like this. So these letters is only 1,000 up to 200,000 base pair in terms of the plasmids. So some placements are present in very low copy number per cell, a few copy per cell. Most others, they do not present in hundreds of copies per cell. So the division, how they divide is also varies, yeah, bacteria to bacteria. Okay. Okay. So now let's me talk little about the central dogma. How does it happen? So as we learned in the previous lecture, what happened? The genes usually encode proteins. So despite these structural proteins or enzymes or regulatory proteins, so in bacteria, actually, the gene is transcribed, the transcription, yeah? They transcribe into messenger RNA also, or mRNA, and this mRNA can be translated into proteins. So this process can occur simultaneously in case of bacteria, as we shown earlier. However, there is more to a gene than just the coding region. So we will start to learn a bit about it, but what controls when a gene is expressed? Yeah, during the central dogma, we say that gene is expressed. How they is expressed? To the polypeptide chain. So all this process in order to make gene to express. So we call it gene expression, right? And how this gene expressed, how highly it is expressed. So this is the question. So this is important. Why? Because not all genes are expressed or need to be expressed during the growth, or so often they might only need to be expressed under certain conditions. So under certain conditions, some genes can be expressed. So the gene expression can be regulated in multiple different stages here. So it can be regulated as the transcription level to the translation level, or even the stability of the mRNA. Okay, this is actually very common not only in the prokaryotic gene expression but also in the eukaryotic gene expression. Okay. So now let me talk about gene expression. What is it? Yeah, it's also very common in the eukaryotic cells. But how does it happen in bacteria, in prokaryotic cells? So as I mentioned, all of the genes in organism usually not expressed. Yeah, some of the genes actually cannot be expressed at the same time, even at the same level. So some gene products that are only required under certain environmental conditions or stages of growth. So some bacteria have evolved a range of mechanisms that they are used to control even gene expression. That's why they are very ancient. They live long life, yeah, and so far they are living. So so that we call it regulatory mechanism are very important um, in microorganisms. Why? Because of the frequent exposure of these organisms to sudden changes in the environment. That is why they have ability to control the expression level. So that depending on the gene and depending on the environment, the regulation could be at one or many of different stages as shown in this diagram. So the first and most important stage of regulation is at the transcriptional level. So the transcription controls when a gene is expressed and how strongly, yeah, it is expressed. In other words, how many transcripts are made from single or several genes, or how frequently the transcripts are produced. So often genes, they are encoding what? Enzymes of a specific pathway. So accordingly regulated, and sometimes one transcript in bacteria can encode multiple different protein gene products. Multiple different gene products. So this is one area where bacteria are different prokaryotes from the eukaryotes, where prokaryotes usually one transcript means one protein product. So immediately preceding the AUG, the start codon, yeah, AUG or initiation codon. Here you see in the slide, we have also what is called ribosomes, binding site. So this is a sequence that is necessary for ribosome to bind to the messenger RNA or mRNA. So to start protein synthesis from the mRNA template, this ribosome binding site, what is called, sometimes called the Shine-Dalgarno. Okay, so you don't need to memorize it, but anyway, so this site exists, and one Australian scientist, he found it. Why bacteria have ability to control the expression, right? So all the secret was actually here in the transcription, how it happens, and in transcription. So starting from the start codon, these sequences, they tend to be highly conserved, which is how they were discovered, highly conserved. But also some of the ribosome binding sites, they are more effective than the others. So here is all this complexity actually depending from each other, but sometimes it can be independent from each other. So meaning that some transcripts can be translated or efficiently more frequently than the other transcripts. So here is irregular things can be noticed in the case of central dogma in prokaryotic cells. Okay. So remember how we mentioned that bacteria can have a plasmid, yeah? Can have a plasmid except for the chromosomal DNA in bacteria. This is an extra copy of DNA with usually just a few genes on it, very few compared to the chromosomal. So this bacteria can share these plasmids. So they are what can I say? Bacteria have this kind of plasmids and they share with each other. They share not only within the same organism, bacteria, they sometimes share with the viruses. Okay, viruses, I mean to say that virus can exploit these plasmids. We can exploit these plasmids. Um, okay, so bacteria can share these plasmids with each other in a process known as conjugation. So this kind of process where they exchange with the plasmids, we call it conjugation. So if the plasmid happens to have a gene that gives some degree of resistance to antibiotic, this may allow the bacteria that receives the plasmid to survive exposure to the antibiotics. That is why. So, which could be very problematic in some point, in some cases. Then, what is the promoter? So, in this case, promoters are the sites where mRNA polymerase initiate transcription at a specific nucleotide sequence, like previously that AGT, yeah, the starting promoter, like that. So these promoters are different in prokaryotes. So the promoters are different in eukaryotes than from the prokaryotes. So in prokaryotes, a single RNA polymerase, this is carrying out all transcription of all different type of genes, whether they have the protein coding genes, for example, the tRNA or rRNA, ribosomal RNAs, etc. So in eukaryotes, the higher organism, yeah, we have specialized RNA polymerase that can be transcribed different types of genes. So RNA synthesis there usually takes place within a locally unwound segment of DNA. We call it transcription bubble. So which is created by the RNA polymerase. So enzyme, because some particular enzymes are involving, right? That is why we call polymerase the endpoint ASE. So the degree of RNA polymerase binding to different promoter, this varies greatly. In this case, in this causes variable gene expression. So another thing why bacteria is controlling the gene expression, the one point here is actually also the promoters now. So this is due to the sequence variation in the promoters. So the mutation in promoter sequences may severely reduce the initiation of gene expression. Severely reduce the initiation of gene expression. So this is how it happens in bacterial genome. Okay. So these are the components of the transcription initiation. So the transcription results in the synthesis of single-stranded RNA molecule complementary to a region along one of the two strands of the DNA double helix. So the DNA in here, the strand that serves as a template for RNA polymerase, it is called the template strand. Yeah, and complementary DNA strand is called coding strand. This is also from the molecular biology. What is that? Uh, when I was talking about the central dogma in eukaryotic cells. So at that time, I didn't separate this is eukaryotic cells, this is prokaryotic cells, but now we are memorizing again. So which strand is coding strand, which strand is the sense, or which one is the antisense, for example. So it is called a coding strand because the sequence effectively the same as the transcript, except we would have used instead of T's, right? Instead of T's. So the initial step in bacterial gene transcription is referred to as template binding. Template binding. Okay. So this side of this initial binding is established when RNA polymerase, we call it the sigma subunit, recognizes specific DNA sequences. We call it promoters. Yeah, in previous slide, I talk about little about promoters. So the promoters are located in 5' region, upstream, we call it, from the coding region. So the point at which transcription begins, it is called transcription start site. Transcription start site. And often when we see the sequence of this kind of gene, this is known as the +1 position. And another position, we have negative minus. So where the transcription usually starts, that is +1 position. So we also have kind of understanding of the operons. Yeah. So the it's a operon model explains how genes are expressed usually. So besides of transcription and analysis, we also have kind of operons. So how gene expression is regulated by the cell? It is actually by the control of the by the what is that? By the control of the operons. So operons are coordinates regulating units of the gene expression. So induction or repression of the gene expression can be accomplished by the same mechanism known as this operon model. Yeah, operon model. Okay. So transcription of one or set of contiguous genes is regulated by two controlling elements. The one element, the repressor gene, encodes a protein, we call it repressor. The repressor binds to the second element, that is operator. Okay. So when the repressor is beyond to the operator, the transcription of the structural genes cannot be uh, occur. Yeah. So whether the repressor will bind to the operator and turning off the transcription of an operon, it is determined by the presence of the absence of the effector molecules in the environment. So all explain how this works in using example of LAC operon, LAC operon. So the now if you want to understand in detail, for example, if someone will ask, yeah, why bacteria have the ability to control the gene expression. So there are many complex things involved, including this operon concept, how the promoters are what is that the starting, yeah, how from the transcription point, how the genes, how what is that the start codon. So all these are mechanism for the gene expression to be expressed in bacterial DNA. Okay, so if you the LAC operon, actually we call it lactose operon. So you heard it about it. The lactose is what is that? It's usually in the milk, right? In the milk. So now let's look at this LAC operon. So the LAC operon of E. coli is given in here. This is consisting of three structural genes. They are Z gene, Y, and A gene. So these encoding proteins and they are called enzymes, plus the promoter and the operator regions. Operator regions upstream of this engine. Yeah, you can imagine this is as one engine. So the number below, the genes in this slide indicate the size and their size in nucleotide pairs, like 780, 825, 300. So we can see here that Z gene is the superior because the nucleotide is big. The nucleotide pair is big. So the LAC operon is one of the best understood operons in E. coli bacteria. So this operon contains three structural genes encoding enzymes that is required for catabolism of lactose, yeah, which is type of sugar found in milk. So the LAC operon, it is inducible operon. So the LAC-Z, LAC-Y, or LAC-A, they are expressed only in the presence of lactose. Only in the presence of lactose, that's why. So the LAC regulator gene, the LAC1 gene, these are encoding repressor proteins. So in the absence of the inducer being lactose, very repressor bonds to the LAC operator sequence, preventing transcription initiation. But this transcription becomes induced by the presence of lactose molecules that is binding to the LAC repressor protein. And this is preventing preventing binding to the LAC operator. Okay, sometimes we see that some Asian Chinese people, they are very sensitive to the lactose, yeah, for example. So these bacteria, maybe they are negatively adversely affecting to their gut bacteria, But this is not the case. But here is the E. coli's the LAC operon, I'm talking about it. So how does it happen? And E. coli is as a model organism. We study a lot of things by just studying E. coli bacteria. Okay. So, however, if, for example, we also have a glucose, yeah? So glucose is present in the cell's environment. That is, the LAC operon will be repressed. So instead of lactose, now there is a glucose. Even if lactose is present, can be glucose expressed. So this phenomenon we call catabolic repression or the glucose effect. It assures that glucose is metabolized when present in presence to other less efficient energy sources. So it is only one it happens only once that the glucose only has been used up that they if the lactose is present and LAC operon will be switched on or switched off, and that is producing enzymes necessary to convert lactose into other sugars that the cell can use it, right? So the cell, I mean, we are talking about the bacterial cell. Doctor, the network is very weak with me. Well, bacterial cell, they like sugar, yeah, a lot. They like sugar a lot. But if you put the milk, what happens? They will convert to sugar. So this is the ability how the gene is expressing. So the catabolized repression of the LAC operon is mediated by regulatory protein. We call it CAP or CAP. Yeah. So this stands for the catabolite activator protein. CAP stands for catabolite activator protein. And the small effectors molecule, we call it cyclic AMP. So they are CAP cyclic AMP complex that must be present in its binding site on the LAC promoter for this operon to be induced. So basically, for this to be the LAC operon to be expressed when both the presence of lactose and the absence of the glucose. So this slide here just show the transcription start to where in the messenger RNA starts of the plus one, yeah, the initiator one, and we can see there is upstream region of the coding region. Here there is start the thymine codon there, and the ribosome binding site would be present just before the methionine, So these are the regions up here, a part of the promoter, including RNA polymerase binding site, the CAP, and the P binding sites, and the operator. So this is how the complexity of the genome of the bacteria, but it's not actually complex as the eukaryotic, because eukaryotic genome is much more complex than what we think now by looking at the bacterial genome. Okay. So now I will talk about some of the mechanism of genetic exchange, because as I said, bacteria can be also genetic, they exchange the gene material, even though they are just like a mitosis, binary fission, yeah? Through the binary fission, they multiply, they replicate. So I will talk some of the mechanism of genetic exchange, how does this kind of things occur in bacteria. First examples we learn about genetics in plants. It was through combining genes from two different individuals, yeah, and these individuals are carrying two contrasting characteristics. One is, for example, dominant, another one is the recessive homozygous, and blah blah. So through how we do it, through the selfing, yeah, selfing. one of the individual through sexual reproduction. So in plants, this occur through pollination, right? And this can be either, as I said, self-pollination or cross-pollination. So this is possible due to the process of meiosis, which results in gametes that they have half the number of the chromosome as the somatic cells of the plant. However, well, how happens in bacteria? Bacteria have only one chromosome and they do not go through the process of meiosis. How it is possible to have genetic exchange then? Yeah, how does it possible to have genetic exchange and also the recombination in bacteria? So there are actually three different mechanisms involved: transformation, transduction, and conjugation. So this is actually leading to genetic exchange in bacteria. So the transformation is the process by which a donor DNA, the male, yeah, in plants, donor DNA molecule is taken up from the external environment, from the outside is coming, right? And it can be incorporated into the genome of recipient cell. So the conjugation, whereas, is the process by which bacterial cells they make effective contact between each other, and then DNA can be transferred from a donor cell to a recipient cell. So in the process of transduction, what happens? This is process by which DNA is transferred from one bacteria cell to another through the action of a virus bacteriophage. So this is usually, we call it it happens in transduction when virus become bacteriophage. When virus become bacteriophage. So we will go on to more detail, and also I have provided some pictures that all viruses, just in a second. Okay, so the first case of transformation, what I say, it is of bacteria. We call it exogenous DNA. They have exogenous DNA. So the DNA fragments from another bacteria enter a cell. So it is possible for that DNA to become integrated into the chromosome of the recipient through recombination. So if these sharing regions of homologue, this is similar to crossing over. So that what happens is between sister chromatids during the meiosis, yeah, in eukaryotes. So in this case here, showing the slide, two crossover between exogenous DNA and the chromosome of bacteria are required to integrate a portion of that foreign DNA into the circular DNA chromosome. So let me see. Okay. So how does then we can represent that crossing over that in order to exchange the genetic material happens in bacteria? So it shows that two crossing over events here leading into replacement of a segment of the bacterial chromosome with the foreign or alien exogenous DNA. And then the endogenous piece of DNA is just degraded. Degraded meaning it is fading away, yeah? So however, if only a single crossover occur between the fragment of linear DNA in the circular chromosome, we can see that these slides here, it would lead to a linear chromosome instead of the circular in bacteria. So this would be unstable and would not be able to replicate and would be also degraded. So this would lead to the cell death. So whether if you had a small circular piece of DNA such as the F factor, which shows some homologue with the bacterial chromosome, we can see that we can get a single crossover event. So that is inserting or integrating this small circular DNA into the overall bacterial chromosome here. Okay, so this could sometimes happen with plasmids as well. With plasmids, this one also happen. What is that? Inclusion, yeah? Inclusion, or this is insertion of some foreign DNA into the exogenous one. So this could sometimes happen with plasmids also. So this process of homologous recombination is also reversible. So we can also get this integrated piece of DNA being excised out of the chromosome. So that is literally meaning we can cut off this space and we can even remove it from the chromosomal DNA, or as well as in the plasmid DNA. Okay, so this slide here is just showing that the process of conjugation. So during the conjugation, what happens? The DNA of an F factor here is the replicated, and it is transferred to the recipient cell that does not have that F minus cell. That is meaning that doesn't have that F factor. Okay, and you can get replication of this F factor here, and it is transferred through to the other cell. So this F factor actually encourages genes that can make the bacterial cell to form this small thin tube. We call it pilus. So this pilus can join one bacterial cell with the another that actually doesn't have this kind of F factor, and then this enables to transfer of the DNA between two completely different cells. So once the effector is transferred into the recipient, so then this recipient cell also becomes F minus, it will become a plus. So it can transfer its effector to other cells. So what you just remember here, there are three types of genetic exchange of the materials. Either it can be through the transformation, either it can be conjugation, or either it can be through the transduction forms. Okay. So now how does this F1, F plus, X to F1 conjugation happens? So one of the best example, and the one we call it recombinant DNA technology. If you heard about it, recombinant DNA technology, we can take the foreign DNA to insert into the recipient DNA. So this technology is using for genetic modification studies. Yeah, this is how bacteria we exploit to have been as a engineer. That's why it is called genetic engineering. So that engineered genetically means to produce human insulin as one of the best example. Yeah, for the diabetics. Yeah, diabetics people. Before creating the insulin, do you know how we get insulin? We get from the animal liver. We kill a lot of animal. We kill a lot of animal, meaning there is a particular animal that is having that insulin. I think, if I'm not mistaken, if it was from the bear. There is a polar bear, white bear, and there is a brown bear. So gallbladder, so some internal organs. We when they are alive, we taken that organ from that bear in order to produce insulin for diabetic peoples. Now, using the DNA, what is the DNA technology, genetic engineering, actually we can exploit bacteria. Synthetic insulin, we call it synthetic, yeah, because it is artificially, we just imitate and we get this product. Okay, so from a person's DNA, we cut it out, person's DNA, I mean, we, not person's DNA, but from the animal maybe, we cut it out and we stick into the bacteria. So we just copy the DNA, yeah, we just copy the DNA using the PCR machine or using the primer, and we insert into the bacteria. So what happens, the bacteria will read it, that gene, and as if it is one of its own, yeah, and it will start manufacture the protein insulin as if it was one of its own proteins. So we are basically using this bacteria a bit like a slave, yeah, to produce human insulin. So step one, in that case, remove the plasmids from bacteria cut. We cut it open with some restriction enzymes. So for cutting the bacterial genome or some piece of DNA, we can use restriction enzymes. So step three, we locate and remove human insulin gene using the same restriction enzymes that we know. And we cut out the human insulin gene from someone's DNA using those same restriction enzymes. Then we need to combine that insulin DNA with the plasmids. So here, plasmids not the bacteria itself, but the bacteria's plasmid we use as a slave. So what to do? We are doing to need ligase enzymes, and they get combined. So step 5, the bacteria are then incubated with the new recombinant plasmid, and they take it up. So the bacteria will then take up the plasmid, and they start to produce the insulin. We can then get that bacteria to produce, and it will pass the gene onto all the other bacteria. Okay, so many bacteria, right? We are not exploiting only one bacteria. There are many bacteria in order to make it in multiplication. So the bacteria we produce millions of times pass on that gene every time, because it reproduces abundantly, and also it reproduces asexually. And very quickly, here we go, we can get a population of billions of bacteria. So this is literally saying we harvest that insulin from the bacteria. All right. Now it is very important part, bacterial viruses. So now I just want to talk about what is bacterial viruses. So the viruses are ultra microscopic, obligate, intellectual parasites. These are parasites. Parasites. We know parasites, right? Parasites. So they are why we call them parasites, because they are capable of autonomous replication within a host cell. That is why they are parasites. So they are not living organisms, but they rely on living organisms in order to what? Replicate. So despite their small size and small genome, they are capable of inflicting a great deal of social and economical damage, and also even to the humans. So I'm sure that all we know that viruses can cause disease in both plants and animals. So some important viral disease of plants we include like necrosis, yeah, the symptoms, how we can distinguish from the bacterial or from the viral. So necrosis, these are, for example, the tobacco virus, cucumber mosaic virus, if you heard about it, cassava brand, straight virus, weight straight virus, and so on and so forth. So there are thousands of different types of viruses that infect plants. Yeah, because we are plant scientists. Often, they multiple viruses can infect the plant all simultaneously. So sometimes symptoms can be mild, in other times they can be very little. So similarly, animals can be affected or infected with many different viruses. So human viruses include also what influenza, polio, coronavirus, uh, what was that, MERS, yeah. So we may not know, but the bacteria can also be infected by certain viruses. So bacteria, that is meaning also uh, what is that? Damaged by the virus. So we call these viruses that infect bacteria, and they bacteria eaters. Bacterial viruses, they're actually bacteria eaters. So what does that mean? That viruses don't literally eat the bacteria, yeah, but they infecting what? The cells. And they take over the cell's machinery for DNA replication, because the bacteria can replicate, they also can exchange the DNA genetic material. Just now I talk about it. So the virus is taking all this machinery work in viruses in order to replicate. So the RNA transcription and protein translation to produce the new bacteriophage particles, yeah, from the beginning, yeah, from the transcription to translation and producing the new protein product. These called virions that can go on to infect the other cells. So the bacteria or bacteriophage, these different species of different types of bacteriophage, can be double-stranded DNA sometimes, but what we believe, so the most of the DNA, the RNA viruses, they are single-stranded. They are having single-stranded DNA, but it is not true. Actually, some of the viruses, they can be also having the double-stranded DNA. Okay, so I don't know if you heard about the bacteriophage that first found how the viruses replicating, yeah, using the bacterial genome. So in bacteria, though, DNA viruses are usually much more common than RNA viruses. So I will give example of few different bacteriophage. One of the most impressive bacteriophage, we call it T4, and this infects E. coli. Actually, this kind of T4 virus or bacteriophage, they will infect E. coli organisms, okay? And we call it lytic phage because it causes lysis of the host cell after infection and replication of the new virus particles inside of the bacterial genome. So it is quite large virus in terms of their size. So it is about 173,000 base pair in length. Yeah. So the T4 virus, these particles consist of the double-stranded DNA, not the single-stranded. And the genome, it is packaged within a head. The genome, because on the picture, as you see, the head is resembling some kind of a geometric figure. Yeah. So this is made up of proteins. So this virus also has a contractile tile, we call it sheath, through which the T4 DNA is injected into bacterial cell. So do you see this is tile pin under? So this because it can inject the bacterial cells like a hypodermic syringe. So the base plate is part of the virus that attaches to the bacterium. Okay. So that is why the shape of T4 is like this. Okay. So let's just look into quickly the cycle of the bacterial T4. How does it come into the bacterial genome? So the whole life cycle of bacteriophage T4, it only takes about 25 minutes. So going from the injection of the bacterial cell through the multiplication of lots of neurons and the lysis of the cell to release those virions. So T4, within 2 minutes, it's injecting the DNA into the bacterial cell, into 2 minutes. So the T4's mRNA synthesis start to begin. So the mRNA molecules of this virus are translated using the bacterial protein for the synthesis machinery. And these proteins then start to replicate on the phage, the DNA. So the phage encode nucleus, this has degraded the host DNA. Okay, the host DNA, it will just attack and it will start, that DNA, that host DNA start to degrade into fragments so that the bacteria can no longer replicate or express its own gene. Very unfortunate. So the newly synthesized phage genome and structural proteins, they start to self-assemble. Assembling means they start to become together into new viral particles. So after about 25 minutes, the bacterial cell lyses, releasing the fully assembled bacteriophage, or another meaning, fully assembled viruses, which can then go and infect other E. coli cells. So maybe it is good virus, yeah, because we are killing E. coli. So then it can repeat the process of the over again. So one infected cell can produce up to 300 virus particles. Okay. So another bacteriophage, we call it lambda. Yeah, lambda, the Greek letter lambda. It can infect also E. coli cells. It's like similar to the T4, but it is also double-stranded DNA virus, and it is DNA is packaged into head also, and also injected into bacterial cell. So the lambda genome is a bit smaller than the T4, approximately 5,000 base pair in length. So one big difference between lambda bacteriophage and T4, it is that lambda is lysogenic phase, but what that means is that through once entering the cell, the lambda DNA or the genome, it integrates into specific site in the bacterial chromosome, okay, and it gets replicated along with their chromosome, and it will start cell division. So in this lysogenic state, it does not cause any harm, yeah, it does not cause any harm to its host. Unlike to the T4, it's starting the host, degrading the host DNA, but in this case, they are coexisting, yeah, they're living. So whether under certain condition it becomes sometimes some certain conditions they will become very dangerous. Yeah, they may degrade host DNA. I mean to say. So in the lytic pathway, the lambda DNA excises from the bacterial chromosome and it will start replicating separate to host chromosome. So it produces a lot of viral proteins and copies of the new lambda DNA genome. So once this happens, the new virus particles are also they join together, like they assemble, the cell lyses and then releases lambda phage into to infect new cells, and this happens abundantly. So there is another type of bacteriophage. We call it M13. This is a little bit different from the previous two cases. So this is quite a bit small. It's about 7,000 only nucleotides in length. In this case, it is a circular DNA molecule, not double strand. But the genetic information of the M13 virus is stored in the single-stranded DNA. So it is single-stranded DNA, the package within the long thread like virions. So the virus enters the cell through the F pilus, which we talked about earlier, okay? And after the code proteins are removed from the DNA replicates via the double-stranded DNA. So intermediate through a rolling circle amplification mechanism. So the progeny of this kind of single-stranded of DNA, they are packaged into new protein codes, and the progeny of the virions are extruded through the cell envelope without killing the cell. So again here, without killing the host cell. So all it really does is to slow down the growth of the cell while it keeps producing the new virion particles. That is why we call it the parasites. Yeah, the viruses, they are actually, and it's essentially meaning they are parasites. Okay, so there is a copies of DNA transfer we call transduction. So what does that mean? Transduction, which is mediated by the viruses. So this mechanism can enable transfer of DNA from one bacterium to another. So during this process of virus replication and host chromosome degradation happens in cases like T4 and lambda. Yeah? So sometimes segment of bacterial genome become packaged into virus particles. So they will start acting like a virus, the bacterials. So instead of the viral DNA, so the spectral DNA can then be injected into another bacterial cell. So it may become integrated by recombination as described earlier. So this one way that segment of genome can be transferred, and this is one way that within the whole bacterial kingdom, genes can be transferred with one another. So even though they cannot cross-pollinate like plants, for example, so the viruses also then they are also part of the whole way in which bacteria can get new genes and also evolving and also involving in new gene function. Okay, so what is the origin of the viruses? It is very, very debateful question. Some people say before the DNA, actually virus was existing. Oh, sorry, the RNA was existing before the DNA. Okay, so where these viruses come from? They're not considered to be living organisms, right? Then how does it? How does they move? How does they choose their target, right? Whoever they have played a major role in shaping the genomes of the both prokaryotes and eukaryotes. So they have also played an important role in our understanding of genetics and gene function. So all viruses depend on the host cell for their function and for their existence. They don't do they do not code for enzymes, they are not involved in energy production. So this dependence on the host strongly suggests that virus has evolved after cell appeared. So viruses has probably evolved from small segment of host cell DNA or RNA that acquired the ability to replicate independently of the regular host genome, which is here in bacterial genome. Overall, we share this planet with so many kind of bacteria that scientists continue to learn about this different type of bacteria, more about their behavior, their changes, their adaptation, right? So if this kind of topic interesting to you, you may want to look onto the study of microbiology. So many careers from agriculture to the medical field to the environmental work, the rest heavily depending on the study of microbiology. Okay, so in this edition, in this type of our last lecture, unfortunately, we are looking at the slides which are posted and listening to this presentation. I would also like you to read the book chapter listed here, listed in the what was that previously, yeah. The principles of genetics. Principles of genetics, there is one big chapter regarding the bacteriophage. For additional information about the genetics of bacteria, if you are really interested, and bacteriophage as well. So thanks for listening. If you have any questions, you can ask me here. Often when this happens in time of stress. Okay. So 1.5 hours. Any questions regarding today's question? Please ask me before we go. Exist this online class. No question regarding what is that? Not quiz. I'm still checking. Yeah, this Tuesday we have the last laboratory practical, lab practical, and I will try my best to announce how you have done in your final quiz.